Look, these products are exceedingly expensive, but I think a lot of the expense, some of the expense is borne by the fact that development costs are really high. Putting a single patient in a clinical trial with a cell therapy costs literally $1 million. I mean, it costs $1 million. A single patient in a small molecule drug trial, so a pill-based drug in oncology, can be anywhere from $100,000 to $300,000, but for a complex cell therapy or a gene therapy, it's about $1 million. So these trials are hugely expensive. You're talking about billion-dollar development programs. So that's part of the cost, no question. But a lot of the cost is also by virtue of the complex manufacturing process. So you think about a CAR T therapy, the way it's developed is you extract a patient's own B cells through a process called leukapheresis. You basically you ship those B cells to a manufacturing site, usually a single manufacturing site, because you don't want to have multiple manufacturing sites so you can maintain tight control over the process. Those B cells get genetically modified using a viral vector, and basically a new snippet of DNA is inserted into those cells to give it the ability to recognize and bind to the patient's cancer. And then that whole therapy is then shipped back to the doctor, who infuses it into the patient. Typically after the patient has undergone some preconditioning, some like mild chemotherapy to partially ablate their bone marrow so that there's room in their bone marrow in their body to take these new cells. And also you lower the propensity or the likelihood of the body rejecting these new cells once they're infused. So that whole process, what we call the vein-to-vein process, is about 3 weeks, what I just described. And it's a very bespoke process that's very expensive. What I would say is that if you look at one thing about the history of drug development over the sweep of 30 years, when it comes to these new biologicals in particular, the one thing that you observe is that the costs always come down, that the manufacturing always improves. These are fundamentally engineering challenges. The engineering challenges always get solved for. People always come up with better methods for manufacturing these drugs, and the costs always come down. If you think about the first monoclonal antibody drugs, so you think about a drug like Rituxan, or Humira, which many of you are probably familiar with. When in the first iteration, the first version of Rituxan was this very bespoke therapy made by a company called IDEC Pharmaceuticals that literally at the time, and this was back in probably the mid-1990s or early 1990s, literally cost about $500,000 per dose to make, because it was a very hands-on, intensive, multi-week manufacturing process with a huge lab infrastructure required to do it. Now we have monoclonal antibodies that are being used to lower cholesterol. That's how much costs have come down, because the technical aspects of making those drugs has improved so dramatically. So I think the same thing's going to happen here with the cell therapies. We're sort of in version 1.0 when it comes to these cell therapies, and you already see technology in the pipeline that would dramatically improve and lower the cost of how these things are made. So we should never assume that the prices that we're seeing today in the marketplace are the prices that we're going to see 5-10 years out, and that the next wave of innovation are going to be priced similarly. I think it won't. But this is part of the challenge of this system that has to absorb the cost of these first-generation technologies in order to see them go through multiple cycles of evolution that ultimately make them far more accessible.